just take a few minutes, but it's my pleasure to introduce Anjali Patel. Anjali is a Springfield native. She's a graduate of Springfield High School. She's going into her senior year at Illinois Wesleyan University, and she's clearly on a steep upward trajectory on her way to medical school. It's been a pleasure to have her in the lab this summer. I can't thank her enough for everything that she's done. I can't thank the donors enough, because this is one of those opportunities where I never really would ever run into Anjali other than this program. I can definitely, when I look at being selective of who I'd like to work with, I usually kind of base it on who I would potentially be working for. And I could see myself working for Anjali one day because she's a smart young lady. So it's my pleasure to bring her to the podium, and I'm excited for all of you to hear what she did this summer.
Thank you. Can everyone hear me okay? Yes. Okay. Okay. So my summer project was on the factors and mechanisms influencing human natural killer cells. So what is immunity? So in your body, you have your immune system, and it's what protects you against disease. And there's two main types of disease that your immune system protects you against. It protects you against infections, so like viral infections, bacterial infections. So for example, if you were infected with COVID, your immune system is what would have been fighting off that viral infection. Your immune system is also able to protect you against cellular transformation, and that usually leads to early cancerous cells. So your immune system is actually equipped to be able to detect these cells and destroy them. However, if they're able to evade your immune system, you can get adverse effects like cancer tumors. And so this knowledge has led to a new line of cancer therapies, which you may have heard of, which are immunotherapies. And so as a result, people have been researching more about the different types of immune cells in our body. So if we take out a drop of our blood, if we kind of zoom in closely, we can see we have a lot of different types of immune cells. We have T cells, B cells, and natural killer cells. And so today we're going to be focusing in on natural killer cells. And so natural killer cells are able to destroy cells infected by bacteria and viruses, but they're also able to destroy cancer cells. And so there's two main types of natural killer cells. You have peripheral natural killer cells, which are generally found in the blood, and they're able to kill infected cells, and they have a lot of anti-tumor activity. And they're CD56 dim, CD16 bright. And so CD56 is like the marker on cells that kind of indicates that they're a natural killer cell, and CD16 is a marker that's generally associated with the ability of natural killer cells to effectively kill infected cells and anti-tumor activity. And so CD56, and so dim means that essentially this receptor is expressed at a low level, and bright means it's expressed at a higher level. But on the other side, you have another class of natural killer cells, which are the decidua natural killer cells, and they're generally found in the pregnant uterus. And so they're very good at promoting blood vessel formation and supporting a healthy pregnancy. And so they're CD56 bright, CD16 dim negative. And so actually this is kind of interesting because actually there's a lot of similarities between the maternal fetal environment and the tumor microenvironment. And they're similar in the sense that, in a maternal fetal environment, you have, cells are rapidly growing because of the fetus is developing. And on the other hand, in a cancer environment, you have this rapid growth of cells as well. And for a healthy pregnancy, you actually need an immunosuppressive environment, which is very similar for cancer tumors, which need to evade the immune system. So knowing this, we looked at the difference between blood and renal tumor natural killer cells. And so there's a lot going on the slide, but if you focus in on the red boxes, you'll see on the chart 2 that says blood natural killer cells, you'll see that there's not a lot of dots, and so each dot represents a cell. But on the other side, with the tumor natural killer cells, you see that there's a lot of cells. And essentially what this tells us is that in natural killer cells in the tumor environment, most of them tend to look similar to the decidua natural killer cells that you find in the pregnant uterus. And so knowing this, we decided to look at the ability of these natural killer cells that you find in the tumor environment to be able to kill cancer cells. And so we did this by mixing natural killer cells and cancer cells in a 1-to-1 ratio. And so what we found was that natural killer cells from the blood are able to kill a high percentage of cancer cells, but in contrast, natural killer cells from your tumor are not able to kill a lot of cancer cells. And so this tells us that natural killer cells from the kidney tumors cannot effectively kill cancer cells. And so really the question here is, why is this happening? Like, what's happening in this tumor microenvironment that is causing these natural killer cells to lose their ability to effectively kill cancer cells? So here's a depiction of your tumor microenvironment. And so as you can see, there's a lot going on. The cancer cells secrete a lot of different factors. But today we're going to be focusing in on 2 factors. So one is hypoxia, which is low oxygen levels, and the other is TGF-beta, which is transforming growth factor. And so TGF-beta is an immunosuppressive cytokine. So essentially what that means is it's a signaling molecule that's able to suppress your immune system. And so kind of the overall research goal is we've noticed that there's this phenotypic and functional change of natural killer cells when they go from the blood to the tumor, and they kind of look more like the decidua-like natural killer cells, but more importantly, they lose their ability to kill cancer cells. And so the goal of this research project was really to identify the mechanism that is causing these natural killer cells to change. And so our hypothesis was that hypoxia influences the expression of TGF-beta receptors on human natural killer cells, and therefore responds to TGF-beta. And so we decided to look at the effects of hypoxia on natural killer cell functionality. And so we did this by taking natural killer cells from your peripheral and culturing them in either normal oxygen levels or low oxygen levels for 4 days. And what we found was that the functionality of these natural killer cells when they're under hypoxia is greatly reduced. So from this, we can draw the conclusion that hypoxia reduces the ability of natural killer cells to kill cancer cells. And so we saw a very similar effect when we cultured cells with TGF-beta for 4 days. We again saw a large decrease in the functionality of these natural killer cells, again pointing to the fact that TGF-beta is able to reduce the ability of natural killer cells to kill cancer cells. And so the question here is, what's happening? How are the cancer cells actually able to cause this change of natural killer cells? And so natural killer cells are actually able to express TGF-beta receptors. And so here you can see, this is a natural killer cell, and you can see that they express TGF-beta receptor 1, 2, and 3. And so here, this chart shows the RNA expression levels of natural killer cells. And so you can see that all 3 receptors are expressed. However, receptor 3 is expressed the most. And then these charts here show also the protein level expression. And they kind of show the same trend, that all 3 receptor levels are expressed. All 3 receptors are expressed at the protein level, and receptor 3 seems to be expressed the most. And so this might be the way that these cancer cells are actually able to cause these natural killer cells to undergo this change. And so we decided to look at the effect of hypoxia on these TGF-beta receptor levels. And so when we looked at the peripheral natural killer cells in healthy donors, under hypoxia, you saw an upregulation of all 3 TGF-beta receptors. And we saw the most amount of upregulation in receptor 2. And then when we looked at peripheral natural killer cells in cancer patients, we saw that same trend. All 3 TGF-beta receptors were upregulated, and receptor 2 was upregulated the most. So since we kind of noticed that receptor 2 seemed to be upregulated the most under hypoxia, we thought that maybe this was potentially the dominant receptor in this pathway. So we decided to look at TGF-beta receptor 2 specifically more in detail. So when we cultured cells under hypoxia, we again saw an increase in TGF-beta receptor 2 expression levels. And we saw a very similar increase in their RNA expression levels when we cultured under TGF-beta. But what's interesting is under hypoxia and TGF-beta, you saw this additive effect of the two causing a great increase in TGF-beta receptor 2 expression. And so this is important to remember because, in your tumor microenvironment, you have a lot of immunosuppressive factors. And you can see that, these are just two immunosuppressive factors, and combined, they can really create a very immunosuppressive environment for these natural killer cells. And so, in conclusion, hypoxia is able to alone induce TGF-beta receptor expression levels. TGF-beta alone induces TGF-beta receptor expression levels, and together hypoxia and TGF-beta are able to cooperate to greatly enhance TGF-beta receptor expression. And so these findings kind of indicate that, maybe potentially blocking the effect of hypoxia and or TGF-beta may restore the ability of natural killer cells to kill cancer cells. And so I would just like to thank all the patients and their families who donated to the study, and also everyone who's contributed to this research project. And I would like to give a special thank you to the SEI Summer Research Program for this wonderful learning opportunity, and also giving me the opportunity to conduct research this past summer.
We have time for a couple questions.
I have one.
Okay.
Since a lot of cancers actually lay down new blood structures, you would assume that with circulating blood you would have more oxygen. So how are these hypoxic conditions in cancer occurring? What's creating the hypoxia?
It's a good question. I'm not sure, but I mean, I would think it's probably because, you have a lot of densely packed tissue. So it's probably hard for these blood vessels to probably move through the tumor environment. But that is a very good question.
Well,
Yeah.
one instinct would be that some tumors grow rapidly. They
Right.
outgrow their blood supply, and so they leave behind dead tissue.
Dead tissue.
They continue to spread out. So that may be one of the ways in which this hypoxia occurs.
Is it correct? Yeah.
Other questions?
So what was the biggest thing you got out of the summer internship?
The summer internship. I think I just really I just really enjoyed research, and I think I just really found that I enjoyed research. And I think one thing I think I learned the most about research is that, you can go in 1 week or even a day thinking that you're going to start doing something, and then your day could totally change because of the results of, the results of what you find. And so I think that's what, I think the biggest thing you learn is that, with research, it's kind of like you're almost searching in the dark a little bit with research. And so I think you have a greater appreciation for it too, I think is what I have, because, it's a hard thing to do, to be able to, continually every day just go in, and you never know what you're going to find, and then being able to actually interpret that. So I think I also gained a lot of skills of just being able to, not only when things go the way that you expect them to go, but also learning from when things don't go the way you expect them to go, and learning what you can also deduce from those results as well.
All right, thank you.